Good afternoon, everybody. So I'm Karen Fallon, and I look after one of the business units at Lonza, and we do all the clinical development and clinical manufacturing, and that covers mammalian, microbial, and chemistry. So this afternoon, as per the introduction, I'm really going to talk about automation and really how we've used that at Lonza to improve our cell line development. So this afternoon, what I'm going to talk about is give you a brief overview of the Lonza organisation, kind of how we fit into that in clinical development. Then really talk about, why we've chosen to go down the route that we have with automation. So we've really looked at setting up discrete islands of automation rather than build one rather large, fully integrated system. So we've done that intentionally, and I'll go through this afternoon and explain why we've done that. And really, then I'm going to talk about how we've used these islands of automation to improve our cell line development process itself. Okay, so just go through the usual disclaimer slide and move on fairly quickly. Okay, so Lonza, the Lonza organisation. So really, what do we do? What do we provide? So we're a supplier to the pharma and biotech industry, obviously, and we really manufacture specialty ingredients as well. So we were founded way back in 1897, so clearly we've been around for quite some time, and our headquarters is in Basel in Switzerland. Last year, our sales, 3.64 billion Swiss, and we are a global organisation. We've got more than 40 sites, and we've got just under 10,000 employees at the moment. So we do have a very large service offering and a very broad product range. And as you can see here, just from this list, chemical and biological active pharmaceutical ingredients, APIs, stem cell therapy, pool treatment, drinking water, sanitizers, cosmetics, nutritional ingredients, agro, high-performance chemicals, microbial control, and wood preservatives. So this is clearly not an exhaustive list, but as you can see, a very wide range of services and wide range of products that we have at Lonza. So thinking about the biologics facilities that we have, so we are a global organisation, as you can see here, the locations of our facilities. So in Slough, in the United Kingdom, mammalian cell culture. Here we perform process development and also clinical manufacturing. Then we move on to our larger facilities where we do more commercial scale manufacturing. So Portsmouth, New Hampshire, up to 20,000 litre bioreactors. Purinho in Spain, where we have 10,000 litre bioreactors. And then in Singapore. In the Singapore facility, we do process development, clinical manufacture, but also commercial manufacture. So really everything from very small scale up to 20,000 litre reactors as well. The Slough site and the Singapore facility, though, these are the two facilities where we really do all of the process development and clinical manufacture. So these are really more the focus of this afternoon's talk on where we have quite a lot of these islands of automation. So in terms of our services and our offerings, as you can see here from this slide, we really get involved with our clients late discovery, right the way through to delivery of drug substance. So, we offer a broad range of services again. So, we can start with our developability services, where we really use in silico and in vitro tools to assess both the manufacturability of these proteins, but also the safety of those. Of our customers' candidates. As you can see, we move through cell line construction right the way through to GMP manufacturing and provide regulatory support. And we supply, very small scale material supply, a couple of grams, right the way through to in-market supply. So the Slough site, as I said earlier, this is one of our key mammalian development facilities. And this is really where the most automated of our processes exist for cell line construction. The the laboratories we have here, the picture you can see, this just shows an example of a typical cell culture lab. They're all identical. We have 23 of those. And we set these up, so we have one lab per project, per customer. And obviously being a CMO, customer segregation is very, very important to us. So, we set ourselves up with 23 identical labs so we can separate our customers out. We do have one isolation lab. This really is there. It just means that we can start some of our customer programs a little bit earlier, keep to their timelines, whilst we're waiting for some of the testing results to come in. So it's really just gives us a bit of an advantage on speed and can bring our customers in a little bit earlier. As you can see here, we also have 2 bioreactor labs, so fairly large bioreactor labs. Again, both the same, and they've both got 45 times 10 litre bioreactors in there. And then we move on to the automation labs themselves. So a whole mixture of equipment in these laboratories, the FACS, automated colony picking, deep well plate selection, etc. And then also our mini bioreactors. And we actually use the Amber systems. So we've got 2 of those right now, and then we're pulling two extra ones, so we'll go up to 4, and that's in Q3 of 2015. So these automation islands and archipelagos that I've been talking about. So I'm now going to really move on and talk about the approach that we've taken. Why have we set ourselves up this way? So as I said earlier, we've really taken the approach of having these islands, and what we mean by that, these are very discrete standalone activities, and then we link those discrete activities together to form this archipelago. And we've taken that decision really for many reasons, operational reasons, compliance reasons, etc. And I'll move through that and explain that to you. So, starting off though, what really are the requirements for a good automation platform? What do we feel that we need, and why we've created these things? Well, we need to deal with non-linear workflows, and what I mean by that is we need to perform certain activities in parallel. Clearly, this saves us time, but it also allows the same pieces of equipment to be used in different parts of the process. So it's really also about flexibility. Having these discrete islands in the process makes it more resilient and more robust. And this is because if individual steps along the process have problems, have issues, we can just remove these isolated pieces of automation rather than destroying the entire platform. So we can keep going just by removing these isolated pieces of equipment. The smaller islands, they also allow us to change out individual components more easily. This allows us to maintain regulatory compliance and also implement any scientific improvements that we want to do. So having these discrete islands makes it much easier to swap things in and out rather than having one great big machine. It also increases our flexibility. You can introduce capability into the system faster. So, for example, if we wanted to introduce an additional screen, we can put that into the system very, very simply. But also, if we wanted to screen cells at an earlier stage, it's easier to put that into the process. Obviously, it reduces error. Having automation, less human errors, and they're resilient and robust. This is the idea why we've put these things in place. They're more resistant to minor changes in operational ways of working as well. And you'll see here, we're talking about 3 archipelagos here: cell line construction, process evaluation, and purification development. But in the centre, these are totally linked by our analytics. So we have very automated analytics in the processes as well to support these. So building these archipelagos. So as I've said, and again, these islands are discrete automated systems, standalone activities. We've pulled them together into these archipelagos, and we link all of these individual items together. I mean, the pictures that we show here, as you can see, these are the individual systems, and they're linked together to execute a major set of individuals. Now, these individual islands can be in different laboratories. They're not in the same place. We have them sited around in different development laboratories, but they're linked by the actual transfer of materials themselves, but also the information that moves from one island to another. So in the as you can see on the top, the photos on the third photo along, I mean, this is just where, the equipment that measures the cell concentrations in an automated system, and the results are used to set up picture number 4, which is really just a robotic island that is preparing the deep well plates for us. So they're linked together by materials and information to set the next one up. And here we've got 3 archipelagos. They're in different development areas, and again linked very tightly by the analytical capabilities that we have. So what I'm going to talk about in a little bit more detail is actually the cell line construction archipelago. Now, if you look at obviously some of the islands of automation that we have, just from these photographs, clearly, there's FACS, there's automated cloning, picking, deep well plate subculturing, and the miniature bioreactor system, so the AMBER. That just gives you an idea of some of these discrete automation islands that we have. Clearly, it's not the full picture, but just to give you an idea of what we mean when we talk about these islands. Now, this has been an evolution for us in Lonza. Up until about 6 or 7 years ago, we had very limited automation. So, we've started there, and we've actually started in the cell line construction process as well. So really, it started off, and it was comprised of just a single automated imaging platform. Obviously, that's what the photograph here is. And we've really grown this over the last 6 or 7 years with plate handling, liquid handling robots, the miniature bioreactors, the AMBER systems that we use. And then obviously really put a lot of time and effort into building the analytical islands around that to support the process development. So pre all of this automation, so 6 or 7 years ago, this was our standard cell line construction process, and really before we put everything in, you can see from this diagram up on the top line, it was really just automated imaging. That was really all we had in the process at this point in time. And really the rest of the process was pretty manual. The overall timeline, it took us about 23 weeks to get to the final lead cells. And really what this meant is we actually had four rounds of screening, so quite a labour-intensive process, and it did take us 23 weeks. Then we moved on to a more semi-automated system. Now, 2 years ago, we actually launched our next generation GS system, so that's called the Exceed technology. So using the Exceed system and some automation, we really increased the number of cell lines that we could generate and that we could screen through that process. This enabled us to reduce the rounds of screening down to 3, so we knocked one of those off, and it reduced our timeline down to 17 weeks, so down from 23 down to 17 weeks. So, as I said, it enabled us to reduce the rounds of screening down, a fairly nice ability for us to actually reduce our timelines down. But because of the automation, it's allowed us to change the way that we're doing the cell line construction process as well. So really during the productivity assessment in the first round, so you can see the circle labelled number 1 here, it really allowed us to increase the number of cell lines that we were processing at this point from 200 to 1,000. We also were able to subculture many more cell lines. So, we've gone over to the 500 area here, moving through the process. And then when we moved on to a second productivity screen using a fed patch culture, typically, again, we've been able to increase the number of cell lines that we've been able to look at, and we've gone up to, again, 500 here. So when you look at circle number 2 on the abridged fed batch, we're enabled to screen and look at many more cell lines. And then we've moved on, and you look at the fed batch productivity in the full bioreactor screen. We're screening about 9 cell lines, on average around here using the AMBER system. So basically, before we had this level of automation, really we were just whittling down the number of cell lines going through the process and moving fewer and fewer along to the next stage. Automating this process has really allowed us to look at much more, many more cell lines, and we're not limited anymore by the numbers of cell lines that we can look at for our clients. Then really looking at a fully automated process. So our current cell line construction workflow using what we call our light path offering. This is even more flexible in its design, and it's enabled us to really reconfigure the islands, and this has allowed us to incorporate the miniature bioreactors at an earlier point in the process, the AMBER system, and this has allowed us to reduce the rounds of screening down to 2, and it again very much shortens the timeline. So we can get down to 12 weeks, so from 17 from the semi-automated process to the point where we can get our lead cell lines for our clients. So as you can see, we've gone through a real evolution. We've been adding automation to our cell line process over the last 6 years. So we've gone down from 23 weeks down to 12. And that's purely the way that we're using these islands and the way that we're improving the flexibility of the cell line construction process. So, automation and the impact really on the cell line construction. So, it really has enabled us to reduce the timeline from transfection to the final cell lines, the final lead cell lines, and that's taken us down to 12 weeks. As I said, it really provides a robust, resilient and flexible approach. And again, it enables us to switch new technologies in very simply and enables us to very quickly keep up with regulatory requirements. It allows for individual changes to the components. So we can swap, as I said, we can swap things in without impacting the full process flow. And it's really enabled us to increase the numbers of cell lines that we've been able to screen. So we've gone from really, a whittling down process and feeding fewer and fewer cell lines through to the next stage to being able to look at the product quality characteristics and the cell line performance on many, many more cell lines. So to enable us to get a better quality lead cell line for our clients. And use of the miniature bioreactor systems as well. Obviously, what we're doing here is we're selecting the cell lines that really are pre-screened to work in our commercial platform process. So, what does this mean? What does this mean to our clients? Obviously, the aim of the game here is we're shortening the time to clinic. We want to be able to identify the most productive, the most commercially viable cell line for our clients. But not just that, obviously one that achieves the desired product quality characteristics. The analytical screening that goes behind this is very, very important. As you can see, we've got these 3 archipelagos very much interlinked with the automated analytics, and you can't screen these numbers of cell lines effectively if you don't have the automated analytics to go behind that. So, and it's also the analytics have enabled us to really are very compatible with many different types of molecules. And it enables ultimately the cell line construction process. We can adapt that, we can be flexible, and we can modify that to meet our customer needs.